What is cancer? It's a question that, in this day and age, most people can answer. Cancer is a disease where mutated cells begin to divide uncontrollably, creating tumors and slowly killing the body. But to some of those people, there's a different answer. One that's much less clinical and much more personal. Cancer isn't just some faceless disease. It's a leading cause of death worldwide, and experiences with a cancer diagnosis can have a devastating impact on one's life. In 2020, the National Vital Statistics System reported that cancer was responsible for over 600,000 deaths in the United States. And a CBS poll in 2017 showed that around 54% of Americans knew someone in their immediate family who was diagnosed with the disease. To those people, cancer is more than just a mass of rogue cells. It's a haunting, looming specter. A specter that does not go unnoticed in research. Over the course of 2023, the NIH has put over $8 billion worth of funds into cancer research, a number that has steadily gone up over the years, but many Americans want to see go even higher. Because even $8 billion worth of investment is not enough to see immediate results from cancer research. Oncology as a whole is an insanely complex field, because the very nature of cancer makes it an insanely complex disease. Many random compounding mutations in varying cells of the body can result in a tumor, and each variation of cancer can demand a different treatment plan, a new drug, a novel therapy. For researchers, cancer is an unpredictable mess that can only be studied little by little, case by case. But there's another somewhat unorthodox answer to this question. Because what is cancer to an oncologist? This is a Tasmanian devil with an unusual condition. Clearly, a large growth is forming in his right cheek, which could be caused by infections or injury. But this one's cancer. It's a facial tumor. A facial tumor that, when it was first discovered in 1996, many Tasmanian devils also developed. What a coincidence, the researchers must have thought. Maybe this warrants some further investigation. And when that investigation settled, something was wrong. These tumors weren't just similar, they didn't just look the same, they were the same. Down to the very genetic makeup of these cells, each tumor shared the same chromosomes. This tumor, aptly dubbed the devil facial tumor, was a novel infectious cancer. A cancer that originally grew in one host, but could spread from animal to animal, feeding off the blood and nutrients of its new hosts. To ecologists, cancer was now a living, evolving parasite. The idea of an infectious cancer might seem strange to you, or maybe it doesn't. Because going by what we said before, you might be able to guess just how all of this works. Remember, cancer is just a mass of mutated cells that divide uncontrollably, and your body is made up of trillions of cells, each of which containing a copy of something we call DNA. An extremely long molecule that encodes the information necessary to carry out bodily functions. When a cell wants to carry out said functions, it chooses a segment of your DNA, a gene, that transcribes into something known as mRNA, a temporary copy of the selected region, which it then goes on to be translated into something known as a protein, a molecule that will actually eventually help carry out that process. This is known as the central dogma of biology, and all of it entails one major thing. DNA denotes function. The information encoded by DNA is what's going to produce the function, and so mutating that information is going to mutate the function. If a part of a cell's DNA, say, encoded for cell division, made a protein that triggers the event, then mutating that piece of DNA is definitely going to mutate the function, maybe causing the cell to divide too much and thus becoming cancerous. Looking past its infectivity, devil facial tumor, which I will be calling DFT from now on, is a truly classic cancer model. When cells are ready to divide, DNA is condensed into large fixed clusters called chromosomes, which can be isolated into an image called a karyotype. This is the karyotype of a normal Tasmanian devil. It has 14 total chromosomes, 7 of which are inherited from a devil's father, and 7 from its mother. This is why 12 chromosomes look like 6 pairs of the same chromosomes, and why there's 2 sex chromosomes, X from its mother, Y from its father. This is the karyotype of a DFT tumor cell. It's completely mutated. You can see the loss of full chromosomes 2, 6, X, and Y, the shortening of chromosome 1, and the addition of 4 uncharacterized marker chromosomes. The DNA of a DFT cell is highly mutated, and surely some of those mutations are responsible for the cell's cancerous properties. But since this is a transmissible cancer, this heavily mutated genome across all known DFT tumors is exactly the same. They all come from the same original tumor, and thus they're all the same tumor. And that's the extent of the disease. It's just a cell whose function to divide has mutated. So while the original mutation can often be caused by an external source, like a carcinogen, hypothetically, all it takes to start one tumor is one mutated cell. So if we found a way to just take some mutated cell from one animal and graft it into another, we would have a transmissible cancer. A devil is expected to die within 6 months of initial infection, which actually creates a recipe for disaster, as DFT is a disease that is both incredibly lethal and incredibly infectious. A devil may die within 6 months, but they'll start developing friable tumors within 2 to 3, leaving plenty of time for an infected devil to spread their tumor. Since the first reports of DFT in 1996, the local population of devils in Tasmania has dropped by about 60%, but its prevalence in this slimming population has not gone down from an estimated 50% or higher due to this property of high infectivity and high lethality. This is made significantly worse by the fact that I've been lying to you. I haven't been talking about DFT, I've been talking about DFT-1. Tasmanian devils have a second clone of DFT with a vastly different genetic makeup that comes from a completely different devil ancestor, showing that transmissible cancers may not be as rare as one might expect. The combination of 2 different infectious tumors actually threatens the species with extinction, something that almost never happens with host-specific diseases. But how does it even get this bad? All living things on this earth are subject to a thing called evolution. It's a concept you're definitely familiar with. Humans for monkeys, that sort of thing. But there's a trick to evolution that isn't obvious at first glance, one that completely recontextualizes cancer. And it starts with the fact that evolution, as a process, is shockingly crude. Quickly, here's how it works. Let's take a population of animals, say humans, which have massive genetic diversity due to mutations and recombination, many different traits across many different humans. When a human reproduces, they pass a copy of their DNA to their offspring, thus imparting their traits onto their child. If said trait makes a human more likely to reproduce than other humans, more children in the next generation will have that human's DNA than in the previous generation, and those traits will represent a higher proportion in the population. As you might expect, this could cause a feedback loop, where said trait begins to dominate more and more of this population until it fixates as a mainstay feature in the species. Natural selection is certainly powerful, but the truth is, it's nothing more than a measure of reproductive fitness. Anything that reproduces and has variation can undergo evolution, which definitely includes cancer. In fact, cancer on closer inspection is the perfect evolutionary engine. It's defined by an extremely high reproduction rate and huge genetic variation in mutations. In a way, one can think of cancer as a process of cellular evolution.
Your body has created evolutionary safeguards against cancer by regulating the division of cells. But because of how evolution works, bypassing those safeguards is actually evolutionarily beneficial for your cells. Whichever cell divides more will inevitably represent more of the local cell population, and when it shows up, that cell will always be cancer. The evolutionary theory of cancer has changed our approach to cancer treatments, which I will absolutely talk about later, but for now we must go back to the topic at hand. Cancer is an evolving entity with all the right tools to become something more than just rogue cells. The more a tumor grows, the greater chance it is for one cancer cell to find the right mutations and break the evolutionary barrier to become transmissible. The only question now is, what do those mutations look like? What does a cell need to do to become a parasite? For that, we need to look at a much more experienced parasitic tumor. DFT has only existed for about 30 years or so. The one we're about to see has existed for over 11,000.
Behold, the humble dog. A simple creature. Everyone knows it, but it's certainly hiding something. Because behind that simple, adorable exterior lies something infinitely complex.
Dogs are one of the 3 animals on Earth that carry a transmissible cancer. If you're curious, the third one is the Syrian hamster, because nothing good ever happens to hamsters. If you go to about 90 different countries, or practically any location with free-roaming dogs, some unlucky dogs will be carrying a shared cancer, known as canine transmissible venereal tumor, CTVT. It spreads in a manner very similar to the devil's tumor, but it's different in one particular way. CTVT is a tumor of the canine genitalia. When the tumor gets large and brittle during 2 dogs' coital tie, a tumor can be dislodged from one dog's privates to the others. A simple strategy, but it works. We know it works very well by now. It works so well, in fact, that this original tumor, originating from a simple sunburn on an old Native American canine, they traced it back 11,000 years. For a cancer to live this long, to become this widespread, one can only imagine what traits it had to have evolved. And from its genome, one that has stood strong since the day it first mutated on this earth, we can read a multi-millennium story that tells us everything we need to know. It's a story that starts with something simple, but ends up infinitely complex.
For a normal cell to make the jump to cancer, it needs to bypass the genetic safeguards that exist to prevent overgrowth. The primary safeguard is regulating the time and frequency by which certain genes are expressed. Remember, the information encoded in your DNA controls a function, and segments of your DNA called genes will induce certain functions when they are expressed. If there are genes that induce, say, cell division when expressed, their expression needs to be controlled. And so, of course, there are genes that do the controlling. MYC is a family of genes that encode for a protein called a transcription factor. Remember that part of expressing DNA is transcribing a temporary mRNA copy from it, so that it can be translated into that functional protein. MYC's expressed transcription factor tells the cell when to transcribe cell division genes, controlling when the cell should divide. But MYC itself will only be expressed with very specific signals, thereby adding an additional layer of regulation to the timing of cell division. Unfortunately, it's at this very mechanism that cancer can emerge. One way DNA can be mutated is via something called a rearrangement, where a DNA sequence gets moved to another location, inserting it somewhere else. And one important sequence in such rearrangements is something we call the enhancer sequence, a sequence that recruits transcription factors to its location on the DNA, telling the cell to transcribe a nearby gene. If DNA is rearranged so that a new enhancer sequence is placed near MYC, transcription factors can cause MYC to be expressed way more often than it should, causing cell division to happen way more often than it should as well. In CTVT, this characteristic rearrangement occurs, and MYC in this case, which we can call CMYC, is now classified as an oncogene, a gene that has the potential to cause cancer. CMYC, along with many oncogenic transcription factors of its kind, is a highly relevant target in many cancers, even among humans. It's a key mutation in the genesis of Burkitt's lymphoma, and a common mutation in a variety of cancers, from breast to colon to melanoma. Developing an oncogene is a crucial stepping stone in cancer's evolution, because it breaks a cell's reproductive limit. Genes that regulate cell division by stimulating it at the right time can be often proto-oncogenes, and identifying them is an important part of identifying potential cancer-causing mutations. The opposite of an oncogene is a tumor suppressor gene, genes that regulate cell division by inhibiting it at the right times. Cyclin-dependent kinase inhibitor, CDKN2A, is one of said tumor suppressors, and as the name suggests, it inhibits the function of a protein called cyclin-dependent kinase. To put it simply, cell division can be broken up into checkpoints, at which a cell checks whether or not it is ready to progress to the next step. This cycle relies on a cyclin binding to a cyclin-dependent kinase, which then activates other proteins responsible for continuing cell division. But what if a cell isn't ready? What if there's something wrong? What if the DNA was damaged during replication because a rearrangement has happened to a special gene and it's too dangerous for the cell to divide? Well then, with the power of a CDK inhibitor, we can simply halt the cell at this checkpoint, repair the DNA, and if that doesn't work, just kill the cell outright in a process called apoptosis. We can see how such a gene suppresses tumors, because it can directly counter the mutations that cancer needs. So how does CTVT solve this issue? Well, that's simple. It just deletes the gene. If the CTVT genome doesn't have CDKN2A, it has no tumor suppressor, so no suppressing tumors. Okay, I think we've established how CTVT has come to become a tumor, but now it has to do something much harder. It has to bypass the host itself. Cells aren't just restricted by your genetics, they're physically confined to a space they occupy. We can all agree that your liver should be liver-shaped, and to keep it liver-shaped, your body has to corral millions of individual cells into a specific place. And the easiest way to do that is by simply tying them together. Between each cell in a tissue is something called an adherens junction, a space where proteins called cadherins connect adjacent cells together. This quick fix to the liver-shaped problem in our bodies is the next hurdle cancer has to overcome, because limited space means limited growth. And luckily, it has a solution so common that there's a name for it. EMT, epithelial mesenchymal transition. Complicated name for a pretty simple process. Since cadherins are proteins, they're made by expressing genes, and if we could just find a way to stop expressing these genes, we'll be free from the shackles of the adherens junction. The EMT is defined by a loss of E cadherin expression, and it results in two processes prominent in advanced stage cancer: invasion and metastasis, processes where the now loose cancer cells break off from the original tumor into nearby tissues, or the bloodstream, starting new tumors. If we just take EMT one step further, and put our now junctionless tumors somewhere less secure, like genitalia, we can see how an infectious cancer is not so far from reality. Due to a lack of cell adhesion, CTVT tumors are extremely fragile, perfect to break off and become infectious. There's just one last barrier that stands between CTVT and total parasikedom, and that's survival. When every genetic and physical safeguard has been beaten, and cancer decides it's here to stay, there's one last defense your body can prepare: your immune system. Cytotoxic T cells are a type of white blood cell that is responsible for killing defective cells, from cells infected by viruses to damaged cells to, of course, cancer. It does so by recognizing a molecule called MHC1, a protein that is bound to the surface of nearly all cells in nearly all vertebrates. MHC1 is like a cell's ID, like a name tag. It lies at the surface because it wants to present a piece of the cell, known as an antigen, to the world. Cytotoxic T cells will check MHC1 proteins, and if it recognizes an antigen as foreign, say one of a heavily mutated cancer cell, it will destroy it. Because CTVT comes from a vertebrate, it has an MHC1, and thus it can be detected and killed by T cells of its host. So, if you want to avoid detection, what do? You hide. CTVT cells reduce the expression of MHC1, a trait that many human cancers also share to avoid T cell recognition. With this, not only can CTVT survive comfortably in its host, but it could potentially survive in any host, owing its millennia-long lifespan to a simple stealth trick.
There are many other strange features to CTVT, tiny improvements the tumor has made over the years to maintain its survival. The immune system also contains something known as a natural killer cell, cells whose sole purpose is to hunt down cells that don't have an MHC. But CTVT seems to dodge them too, via a poorly understood process that dampens the immune response of the surrounding environment. CTVT is a highly mutated mess, and while that's amazing for genetic diversity, it should be terrible for long-term survivability. But CTVT's genome is highly consistent across tumors that lie thousands of miles away and millennia apart. It turns out that CTVT could be fixing its genome by stealing DNA from nearby host cells, parasitizing genetic information. And finally, perhaps the bit that I found most interesting, CTVT potentially alters the behavior of the dogs that carry it. The tumor increases the expression of estrogen receptors, which could change the estrous cycles of female dogs, possibly encouraging the sex behaviors that causes CTVT's spread in the first place. What should have been a simple tumor has turned out to be a mind-altering, gene-stealing parasite. But that's enough. I think it's finally time to leave it at that, because with all of this in mind, we now know what these cancer parasites are, how they operate, and how they came to be. And all that's left is to answer one final question. What can we learn from them?
The first way I could wrap this all up is by telling you how researchers use this knowledge to actually combat cancer. One thing you've probably caught on to at this point is that many of the adaptations transmissible cancers use are also used by actual human cancers. And that's not a coincidence, it's just how cancer works. Due to a lack of genetic safeguards that would normally repress mutations, a tumor population will have incredibly high genetic diversity, and that massively increases its speed of adaptation. This means that any tumor can contain, say, drug-resistant cancer cells. And when we use a drug to treat that tumor, we're just going to leave those resistant cells alive. In doing so, we've actually made the problem worse. By killing the surviving cells' local competition, we've given it more access to the nutrients and the room it needs to expand, leaving us with an aggressive, drug-resistant tumor. This fact is playing a role in how we approach cancer treatments, as therapy has shifted from single drug therapies to multi-drug treatments, bypassing potential resistances. But we can apply evolutionary theory in different ways to counter this threat, perhaps by designing therapy plans that select for weaker cells first, or using lower continuous doses to slowly kill a tumor while keeping its competition alive. The ultimate application of evolutionary theory, however, is by recognizing that the most critical period for a tumor is when it first appears as a neoplasm and begins garnering genetic diversity. How early a tumor is detected already plays a massive part in a patient's survival, but now we know that the earlier we can catch a tumor, the less capable it is of adapting to our treatments. In this world of evolving cancers, improving cancer detection is as important as finding new cancer treatments.
The second way I can wrap this all up is with something completely insane. In my hands is a hypothesis written by Russian biologist Alexander Panshin. From what I can tell, he's a trustworthy scientist, but he's mostly known for running a science-forward YouTube channel. So you know he's a bit of a sensationalist. But bear with me just a moment. You've stuck with me for this long, so let me tell you about the scandal hypothesis. Basically, there exists this multicellular organism, Henaguia salmonicola. It's a parasite of fish. It's this little white spot over here. And it's technically an animal. It can be traced back to hydra and jellyfish ancestors in phylum Cnidaria, but it's obviously much simpler than its ancestors. So simple, in fact, that it violates one of the basic implied laws of biology. It doesn't need to breathe. Every animal needs to breathe. Every complex creature uses oxygen. Cellular respiration is the most efficient way to make energy. But this parasite doesn't need it, because according to Mr. Panshin, it didn't just evolve from cnidarians, it evolved from cnidarian cancer cells. Life, actually defined life, could have evolved from cancer. The evidence is simple. This species has lost a lot of genes, the big one being a tumor suppressor, caspase. Caspase expression is responsible for apoptosis, a cell's way to self-destruct when things go wrong. Absolutely mandatory function for any multicellular creature. After all, you don't want to risk a cell becoming mutated and messing everything up, unless you're cancer. Add this to the fact that many cancer cells don't use much oxygen. They obtain energy via a different pathway called fermentation due to something known as the Warburg effect, and we have a theory on our hands. Just imagine, if you will, you're a fish harboring a nasty parasite, and that parasite gets cancer, and then you get cancer, and then you transmit your tumor, like devils and dogs did before it, to a fellow fish. And it transmits from fish to fish long after you're dead, mutating and gathering so many different kinds of random genetic garbage that you eventually become multicellular, and you become an animal. And you're alive. Crazy? Yeah. Impossible? At this point, I don't think so. At the start of this video, I asked a seemingly simple question. What is cancer? And now that we're finally done with this mess, it's time you ask yourself, how has your answer evolved? And with that awful closing statement, I just want to thank you if you've actually stuck around this long. This has been a pet project of mine for such a long time now, and I've just basically stuck around with this idea for so long that I didn't think I would ever really finish it, but I decided to push for it right now, and it's been a wild ride. Clearly my editing experience is not very high, but I really hope that if you stuck around for this long, you've been patient enough to see through it, and I hope you really have enjoyed it. I hope you've come out of this video learning something about the wild world of cancer, and yeah, I really want to make more of these in the future as well. I want people to learn more about the molecular mechanisms behind our world, and so if you're interested in learning more, I'm planning on doing another video in the future, actually, specifically on the mechanisms of fibrodysplasia ossificans progressiva, a genetic disorder where a person's flesh slowly turns into bone. So If that's something that excites you, if you like this style of video where you take a sort of deep dive into molecular concepts, into biological anomalies, I don't know, I guess subscribe or just keep on the lookout for something else like this. I don't know. Again, though, thank you so much for sticking around. This closing statement was a mess, but I hope you've really enjoyed. See ya.